Our next speaker is Dr. Marachelian. She is the director of the neuroblastoma program at Children's Hospital Los Angeles. She is also an attending physician in the hematology, oncology, and blood and bone marrow transplantation group, and at Keck School of Medicine at UFC of CHLA. She is a California-trained UC Berkeley undergraduate, and she did received her medical degree at UC Irvine and her fellowship at Children's Hospital Los Angeles. She is also the chief investigator at the New Approaches to Neuroblastoma Therapy. She has also obtained her master's in clinical investigation at USC. She serves as the medical director of NANT, the NANT Consortium, since 2010. Her work in translational research focuses on neuroblastoma with an emphasis on immunotherapeutic approaches. Her expertise has allowed her to lead multiple clinical trials in this field. She's a member of numerous organizations and associations, and obviously author and co-author of many articles and publications. Her talk today is going to give us a little bit of history on the NANT and what it is and what the current trials and results have been over the years. Dr. Araz Marachelian.
Right. Thank you so much for the invitation, and I hope to be able to switch gears a little bit. I'm really going to focus on relapsed refractory disease, and we'll talk about what has happened with NANT, what is NANT, what does it do, and try to tell a few stories about how our therapies evolved and the rationale. So, first of all, everyone's got a vision, and our vision is to cure all children with high-risk neuroblastoma using novel precision therapies developed by our expert network of laboratory and clinical investigators. What does that really mean? There we go. This is an old picture, actually, of one of our NANT annual meetings. You can see this is a group of individuals who work together. It has changed a little bit and evolved, but in the center, if I think I have a mouse, but in the center, you can see some of the original NANT individuals who started off NANT. But the concept was, we need to work collaboratively. Really, neuroblastoma is such a rare disease that if we all do trials on our own, we are not going to make as much progress. And so bringing minds from various disciplines together to achieve goals of improving outcomes and providing access to patients. You can see there have been quite a few patients enrolled over the years, including more than 800 therapeutic studies. So ways to access for patients for clinical trials while we are learning how to make things better. I'll just talk about what a program project grant is, and that's because NANT is actually partly funded by the National Cancer Institute as a funding mechanism. The concept of a program project grant is it's supposed to support a broad research program. And it's supposed to have a major objective or theme, and in this case, it's neuroblastoma therapy. It's supposed to be broad-based, multidisciplinary, and long-term. And the idea is for the National Cancer Institute to provide some funds that are a little longer than 1 or 2 years so that there's stability and people can crosstalk and do the research. So this partially funds our consortium. It funds the laboratory of the research program as well as NANT. NANT is the translational core, meaning it is the one that's translating what's in the laboratory to the patients. The goal is to discover and exploit mechanisms of resistance and immune escape. You can see the centers that are involved with this grant. It used to be Bob Seeger who started this, and currently the PI is John Maris at CHOP. with the operations at CHLA. Myself, St. Jude is where Julie Park is, and she's the scientific lead with me in the NANT. And the other groups, in addition to CHLA labs, are the UCSF, the DFCI, and Stanford labs. There we go. So here's just this idea of what are we doing. We're trying to leverage the consortium, clinical, biostatistical, and operational infrastructure to accelerate the translation of the new therapies and target vulnerabilities in resistant neuroblastoma uncovered by the laboratory projects. And so we have the infrastructure for the rapid implementation, and then also, though, have a uniquely annotated biobank of therapy-resistant neuroblastoma patient samples and biostatistical support for correlative analysis so that we can then go from bench to bedside and back. So here is the broad view. You have these investigators working on the treatments and the preclinical testing in the laboratory. And then you design and carry out some early phase experiments. The clinical investigators are involved in that clinical decision-making in the preclinical studies. We have our NANT sites who enroll the patients, collect consistent data with good quality, and provide the rationale for larger studies then in the Children's Oncology Group. And then via this whole mechanism, also offer patients access to novel therapies across a broad geographical area in North America and Europe. An important factor of all this is that we have a parent advisory committee, many individuals you may have heard about and who are involved in our parent advisory committee. We integrate their input in the early phases of our development throughout, and we have started embedding them in each one of our protocols, and so as well as the program project grant. So it's been a really interesting evolution and a beautiful collaboration. So here we go through the NANT sites. There were 8 original sites. It's about 18 currently. Different trials have different individuals involved depending on where the drug can be shipped to, but of course it's geographically chosen so that we can have access for patients and for quick translation. And you can see it's not exactly beautifully spread out, but it is trying to be spread out. And on the left is the NANT headquarters at CHLA, where I work. So next slide is showing our European group. So we did have Europeans collaborate with us in our prior studies. We have expanded that a little bit with Princess Maxima having an operations now for NANT as well, so that we can more quickly collaborate with our European colleagues so that we can do transatlantic studies when possible. In addition, we have Toronto Sick Kids, who has always been participating in our trials when, again, we have access to the agents. So how does it really work? I only put this picture because I feel like it seems like it should be a straightforward line and hopefully going up, but it is actually lots of up and down. So we have the lab work, lots of collaboration, lots of talking, lots of discussions with industry, and then getting to design and executing the study, getting feedback, and really one of our main problems, getting the agent, even if we thought that it was working in the lab, well, getting it into the clinic is a huge effort and requires a lot of back and forth with industry and regulators. And then finally, if successful, we're able to move it to our neuroblastoma patients in relapse refractory. And if it's good, then we can move it over to usually COG, and in Europe now, the Cyapin. So I'm going to tell the story of a few agents, if I can get this working. So the first story is about, I apologize. about lorlatinib. And the reason I chose lorlatinib as a story to tell is because it just shows exactly how our trials go from one to the next, while explaining a little more about how lorlatinib works in ALK-resistant neuroblastoma. If I can figure out which direction to go. Having some problems with this mouse. I think I'm going back. Oh, that's why. All right, now that you saw a few of my slides, we can go forward. ALK is a mutation. There's a mutation in the kinase domain in neuroblastoma that leads to activation. And there is something called amplification, but most commonly in neuroblastoma, we have actually mutations. And what was really known is that the diagnosis, we have ALK mutations probably in about 10% of patients. But if you look in the relapse refractory population, this is higher. It's about 30%. And so clearly something is evolving there. And it's an area that does need looking into. So a lot of the early work on lorlatinib and ALK was actually in the Philadelphia lab, where work was ongoing to try and figure out what are the best agents to move forward for ALK-mutated neuroblastoma. And although initially there was crizotinib as the early studies, there was work at Dr. Mose's lab that showed that lorlatinib, which was a different ALK inhibitor, was superior to crizotinib. And that work was, as you can see, there's a blue line right there with lorlatinib with chemotherapy. You can see the tumor volume in these experiments was much, much lower, where you can, and so this idea, this lab work that we were seeing was the reason we started the trial in NANT for lorlatinib. So this was for patients who had ALK aberration, so a mutation or a amplification, and we had different cohorts, the younger group, the older group, so the adults, and then we had one with combination with chemotherapy. There was work on blood for circulating tumor DNA collected at intervals, so that as we went along, we could understand what was happening with lorlatinib and ALK and on the other mutations of the patients. So here are some colorful figures of what it looks like for our patients. So you can see the first group there is cohort A1. These are the pediatric kids, patients who were younger than 18 years of age, and they got lorlatinib, and this is MIBG Curie score change. And the color coding is to look at the type of response: complete response, partial response, minor response, stable disease, and progressive disease. And you can see the sad gray is our progressive disease patients. So we did have some pediatric patients who had some tumor growth with lorlatinib despite having an ALK aberration. But you can see that the majority of patients actually had improvement in tumor size, although if you looked at just specifically what you call complete response and partial response, that rate was 30%, was what improved with minor responses. This was different in our adult patients. You can see the adult patients, there was only one patient who had progressive disease and only a slight increase, and all the rest of the adult patients had improvement in their disease. So there's clearly something different in the patients with, who are pediatrics than adults in terms of initial response with monotherapy. What's encouraging is that when we did a group for what we call cohort B2, which is when we combined it with chemotherapy, that response rate improved in pediatrics with the chemotherapy, and you can see it was more similar to the adults. So this whole concept then took the compound from the laboratory to the trial and then translated it over to Children's Oncology Group and now Europe, although Europe is a little bit lagging behind, and this is the reason now patients who are newly diagnosed with neuroblastoma who have an ALK aberration are able to go on the study, the phase 3 study with lorlatinib upfront. And our hope is some years down the line, once we have enough patients to evaluate, we will understand the outcome of the patients who have been treated with lorlatinib even upfront and not just wait for the relapse. So next slide just shows what else did we learn. Yes, it's beautiful that we saw that, but we also need to understand why did it work and why didn't it work. And this is an opportunity where we looked at circulating tumor DNA and looked at patients. There's clearly groups of patients, some patients who are really what we call ALK dependent, whose ALK level goes down, and then as soon as they do progress, it goes up. There's some that when they progressed, it wasn't an ALK mutation that was driving the progression, and in some other patients, we didn't even see ALK detectable in their blood initially. So a lot of understanding, mechanistic understanding of this, so that we can then, this is the part that we have taken it back to the lab, and now we will try and understand a little more of how to perhaps make it work a little bit better in the future. If I can figure out which direction to go here, this mouse, here we go. And you can see this is the kind of analysis that goes on where you can see a patient, these boxes are at different time points. You can see their tumor mutations are changing with therapy. And really the reality is we do know when you try to inhibit one pathway, another pathway sometimes can try and overcome. And this, you can see this heterogeneity, and Dr. Moset's lab actually has taken some of this and gone back to the laboratory and done some more experiments while we're working further. What I can say is reality is what's next here. We'd like to figure out ways to combine this with other novel agents. We are working on that. Of course, again, drug access is an issue, so stay tuned, and we will try and figure out what our next study with, for our ALK aberrant patient is. But this data, the data from the patients has really helped inform that in order to move it forward. Similarly, this is the same concept where all the patients on NANT are, go on our biology study where we can get blood for DNA, RNA, and we can get their immune cells and store them, and any scientist, whether involved in NANT or not, can request these samples, and there's a scientific review so that we can try and understand, again, how to improve this. And when you look at this, we did a precision study where we looked at biopsies and tried to put patients in different categories and say, oh, you're an ALK patient, you have a MAP kinase, different kinds of inflammatory metabolic group. And really, there's a lot of work that will be going on for a long time. But what we can tell for sure is that neuroblastoma is completely heterogeneous. There are patterns, however. There are patterns of, one gene with one chromosome, different ones. And I think part of that, the issue is that that means that each one of these may require slightly different kinds of therapies, which make clinical trialists lose their mind because statistically that becomes very, very difficult. But at this stage, we are learning quite a lot and understanding the disease a little more so that we can figure out how to target the different groups of neuroblastomas in proper ways. So an evolution of the biology and our precision is what we call the NANT Mighty protocol, which you will see come through the beginning of the year. We are currently working on it quite a lot. What is the proposal is to take all patients who are going to enroll in a NANT study, therapeutic study, and really in detail analyze their tumor cell load in their blood. genomics, as well as really correlating that with their response to an outcome. So this is really in the context of a therapeutic study, because we all know, yes, circulating DNA are all things that we have used in our clinical practice. Sometimes we try to make a decision one way versus the other, but we need to do it in a context of a clinical trial so we can really understand as a group how to really be able to use it without opinions, but rather evidence-based. So what's happening here is that we're sequencing the tumors and returning the results, and a liquid biopsy is also going to be sequenced and returned to the patients. But there are other studies, like you heard Dr. Reynolds yesterday talk about GD2, since that is not yet a study that is clear, meaning it can be returned to patients. It's being done, and we're analyzing it and hoping to get enough data so that it can get to a point where we know how to use it and return the results that might be helpful. And doing the same thing for immune profiles. So patients have a lot of different kinds of immune profiles, and understanding that is also beneficial over time. At this point, none of these are actionable, where I can return them and tell the patient, you know, I see this and therefore you need X, Y, and Z, but at least we have those instituted so that we can try and understand this evolution over time while they are getting therapy. So this is the pretty picture. The idea is hoping if we have enough trials and enough agents, and our industry partners want to play, we could start a patient on a study, hopefully have some biomarker-driven trial, then understand, there may be a non-biomarker-driven trial as well, understand, do data analysis, put them together, and then go forward and perhaps go to the next trial. So this concept that this is a dynamic and evolving phenomenon, and we need to understand the interplay of tumor genomics and microenvironment and immunology. So lots of serial monitoring and hopefully some rational ideas going forward, but stay tuned. A lot of this work is going to require many institutions, of course. We are involving Europe in this project, and in fact, it's one of our first big endeavors within Europe, and you can see the type of testing that's going to be done. Some of it that's going to be returned and some of it not, but some of it we expect the results to be much quicker, and some perhaps like spatialomics may be things that might be a little bit further down the line in terms of making an impact for our patients quickly. These type of tests are going to be done slightly differently in Europe, but parallel, so that samples are not going across the continent, but rather in each one of the areas. Again, back to collaborating together and trying to see if we can work together, ask some of the same questions, and put it together. So I think most of you here know the other story is, of course, chemoimmunotherapy that has changed what we do in neuroblastoma, and of course changed what we do in relapse refractory neuroblastoma due to the work that occurred at Children's Oncology Group and the improved outcomes with chemoimmunotherapy. This is great, but what we need to work out is how do we make this better? So part of our work right now involves trying to optimize that. While we're working on this, we also simultaneously established that vorinostat, when added to MIBG therapy, improved outcome in neuroblastoma for MIBG avid disease. And this was a trial that was done through NANT, a phase 2 study led by Dr. Du Bois. The study looked at 3 groups of patients with 3 groups of therapy with irinotecan, Temodar, in one arm, vorinostat in the other, and as well as in the other arm, just MIBG alone, to try and understand whether if we add vorinostat or irinotecan and vincristine, if we can have any improvement over MIBG alone. And so since this study showed the vorinostat had improved MIBG therapy efficacy, the next step was to try and figure out where do we take this next. And I do not know why this is giving me trouble today, but mainly what we decided, obviously we know that not everyone was responding. So in order to understand what that was, is we know there's some resistance to DNA damaging agents. We know there's some downregulation of receptors. We talked about that yesterday with the antigen, the GD2. The tumor microenvironment, which is this big word for lots of other cells aside from neuroblastoma, and of course our host immunity. So how do we tackle all this together, step by step? So the next step was to look at adding an agent. So we just learned about vorinostat and MIBG. But beautifully, during the same time, there were preclinical studies that showed actually vorinostat upregulates GD2 in the cell surface expression, which of course then creates a more permissive tumor microenvironment and therefore can enhance anti-GD2 antibody responses. And so you can see here in the experiments where the addition of vorinostat plus the anti-GD2 was the one with the least tumor growth. And so this was the reason the trial that came after was done for, that was called 1701, which was MIBG with dinutuximab and vorinostat. So you can see the study, okay, here we go. So this, the rationale, of course, we know MIBG is very active. We know dinutuximab is very active, trying to combine them rationally, but also knowing that radiation can change the local tumor immunophenotype and inflammatory response, and then you can have your immune system recognize the tumor better. And then the fact that vorinostat then upregulates GD2, it was all the rationale for the study, and the study then added, here we are, regimen A was the initial combination of MIBG with dinutuximab. A week later, once you're out of your radiation isolation, you have your stem cells, then you get dinutuximab again, and then you get your disease evaluation. And in regimen B, once we identified that vorinostat was upregulating GD2 and the data from MIBG and vorinostat, we added that for 14 days during the MIBG. And you can see then, this is the efficacy data. This data is going to be published very soon. You can see here the first 3 dose levels. These dose levels did not include the vorinostat. Similar to what I showed with the lorlatinib, you can see the change in the MIBG Curie score. And again, we like ones that are going down and not up. Luckily, there are not that many up here, and there's a lot of really nice response in all our dose levels. Dose level 3 had a higher number of patients by design. And you can see once we added vorinostat to this, the response rate really was even better. It's a response rate of 75%. Of course, that includes CR, PR, and what we call MR, minor responses. The reality is, of course, I can't for sure tell you, this is not a randomized that vorinostat added, but enough data to tell us there's some evidence that this could be a path forward and may be beneficial for our patients in combination. And what's more important is the fact that tolerability was very good for both MIBG with dinutuximab and MIBG with vorinostat and dinutuximab. Perhaps with the second course, the vorinostat arm had slightly more hematological toxicities or non-hematological, some potassium, et cetera, but tolerable. So this was the reason, this exciting data is the reason why we have a planned study that is planned to have MIBG with vorinostat, MIBG with dinutuximab, and then MIBG with dinutuximab and vorinostat to try and understand which one of these arms is actually superior. Unfortunately, right now, we are still pending MIBG supply, and once we do get the supply and are able to secure that, we should be able to go forward with the study. So stay tuned. So how do we then improve chemoimmunotherapy? This is MIBG, and we've added agents to it, combined it with immunotherapy, but there are ways to just look at MIBG itself. So the path right now that we are working on or embarking on are NK cell-based immunotherapy, mainly because natural killer cells are good killers of cells, in addition to what I think Dr. Reynolds talked about, neutrophils, et cetera. So our concept here is to see if we can make them better. There are two ways to do that. One is to give better natural killer cells to our patients. The other is to activate them. The NANT 1301 was the first study where we embarked on this, where we took patients' own NK cells, took some blood out of them, sent them over, grew them in the lab, and returned back to them. At that time, this is pre-chemoimmunotherapy, so it was just being given with dinutuximab. And it was feasible, and we were able to do it in a multi-institution fashion, which is very important, I think, for cellular therapy going forward in order for us to make progress. However, the problem was that neuroblastoma cells, NK cells, were a little bit too sleepy and too few. And even though we tried to activate them, we just didn't get enough to think that this is a long-term pattern and us going forward and be able to utilize that concept. So went back to the laboratory. Actually, Dr. Dean Lee, who's at Nationwide, worked on looking at what we call off-the-shelf product. This could be something that we can give right away, we can give more often, and we can have perhaps efficacy if you chose the right donor. If, you know, so not all of us are born with the same number of NK cells or the same potent NK cells. And eventually, if you do this, you could have some decreased costs. And so this was a concept that we all thought scientifically would make sense. I am not going to bore you with the science on this slide, but just to tell you that TGF-beta is something that is pro-tumor, and the tumor actually releases it, makes it work harder. In fact, when you do anti-TGF, it synergizes with anti-GD2 and NK cells, and unfortunately, TGF-beta anti-TGF drugs are not really available to us right now. So one concept has been to educate these NK cells with the presence of TGF-beta so they can be stronger. So what is happening currently is these NK cells from the donors, these are really the best type of donors of NK cells because they have more activating what's called KIRs. They are sent to, they are put in the lab, and they have what I call in TGF-beta school. So are they being grown with TGF-beta? We call this TGF-beta imprinting. That way they're educated to be resistant to immunosuppression. This type of cells, there's some safety and experience in other diseases, and so we have a trial for patients who are eligible with any relapse number, so they don't have to be first relapse or fifth relapse, but they shouldn't have progressed on chemoimmunotherapy before, but because we want them to be patients who could have a potential to benefit from chemoimmunotherapy. So there's chemoimmunotherapy. You can see day 1 through 5, patients are given irinotecan, temozolomide, and dinutuximab. Dinutuximab is day 2 to 5, and then on day 8, the NK cell infusion is given, and GM-CSF is given right after the chemoimmunotherapy. However, if patients are allergic to GM-CSF, they can get G-CSF. This study is ready to be launched in the US, but not in Europe, because there's cellular therapy, and shipping those cells would be problematic at this point. So we have the trial ready to launch, actually hoping next month, and we have some patients who will be hopefully lined up for next month and the following months. This is a phase 2 trial, so we are looking at the response rate with NK cells and chemoimmunotherapy. I talked about how to make NK cells better in bigger numbers, but you can also think about activating them. You can give them something to make them angrier, work harder. And there's some data from St. Jude where IL-15 with chemoimmunotherapy was helpful to allow for tumor regression. And you can see in group 4 that we're all given with that. That was the group that had the most improvement in tumor volume. And so this concept is going forward in NANT sites as a trial. However, this trial, instead of being given with dinutuximab, is being given with the St. Jude antibody, and it is another phase 2 study. Again, chemoimmunotherapy is the base, but this time you have the St. Jude antibody with this N803, which is the activating IL-15. In this trial, it's a randomization to allow for the chemoimmunotherapy versus the chemoimmunotherapy plus the ALT-803, rather than everyone getting the 803, and this is really also because we want to look at the differences between these groups. This study is a NANT collaboration, but is actually being run out of St. Jude. So last but not least, this is the study of chemoimmunotherapy. Here, the next step is really looking at how do we quantify neuroblastoma response. This is an area that we have been working on quite a lot for any parent who has gone through neuroblastoma therapy, especially in relapse. Whether a patient is progressing or not, or whether a patient has is really benefiting from something, sometimes is a little challenging to understand. So we're focusing on looking at our response criteria and trying to understand how to improve them. This resulted in our international response criteria revision. However, we're doing a lot more work on it to look at new MIBG technologies, how to interpret low level of bone marrow involvement, and correlating soft tissue uptake with MIBG, et cetera. One of these days, we're going to get lots more work, probably with some AI help, but stay tuned. So ultimately, we have a lot to still learn. We have circulating tumor DNA data coming through. We have mRNA data, which is another way of looking, I think Dr. Applebaum talked about. We have these data, and we're currently looking at it in context with our therapeutic studies and trying to understand who are the patients who are likely to respond versus not, at the same time as can we predict someone to progress before they progress. That work is ongoing, and we hope to come back in a year or so to be able to see if we can shed any light. I think I may have talked about this last year when I gave this talk. We had a study that was looking at MFBG, which is a PET imaging study. There was a phase 3 study that we just completed that was looking at MFBG in order to look at really the differences between looking at MFBG in addition to MIBG or in comparison. This MFBG scan may have a higher sensitivity. It's a quicker scan, and it doesn't require thyroid protection, so it's sounds like a really good deal. However, it is not yet FDA approved, and so the data for it, we do not know how to still use it. So the preliminary data has been looked at, and we can see some increased number of spots when you compare to an MIBG scan. However, at this point, we're looking at that analysis and trying to understand what does it really mean and when can we use it. Of course, in order to do that, there's two settings this could be used in, one in the relapse refractory setting and one in the children's oncology group in our upfront patients. So patients in the upfront are likely going to go forward with a study where this could be incorporated and looked at, again, as a correlative, not as a way of decision making, because it's still too early to decide about what does it mean if you have a spot at the end of before transplant, what does it mean at the end of therapy? This is what needs to be done, and it is being worked on. We will see. I think some of this work came from actually Memorial Sloan Kettering as well, and so this was a collaboration, and we'll have to see where we go once we get the data on hand. Ultimately, the way we're going to learn is by pooling data from multiple studies, and this is what we are currently doing, whether it's the biological data, all the radiological data, and these are available for radiologists to try and ask questions. We are trying to understand how to improve this field generally. And so all this will help us design future studies and inform the public, hopefully. And last but not least, there are still lots of challenges. As I said, relapse refractory neuroblastoma is very heterogeneous, so it makes for an interesting way to study a disease that's probably multiple diseases in some way. And we do know that we think success is combination therapy, but the learning is bidirectional. So what we are doing, studying the patients and then going back to the lab, but this requires infrastructure, and this is what we have set up so that we can make that happen. And of course, as you can see, it's many, many individuals involved. I'm giving this talk today, but there are many others, and it does include early collaboration with industry and our parent advisory committees. Lots new trials are in development at the moment, but our drug supply access remained our biggest hurdle in getting more trials to our patients, and the advocacy that you all do and we all do, and hopefully eventually will overcome most of these hurdles. And so thank you very much. We have a huge group of individuals at NANT sites, but also our patient and families who have really done so much work and commitment to getting our patients to our sites. And of course, our usual supporters and funders who we could not do without. Thank you.
Thank you, Dr. Marachelian. I have a question there, or no? Oh, yes.
Thank you so much for coming and presenting. I was wondering why vorinostat was chosen. I think you had a slide on it, but could you tell us kind of how vorinostat works?
Yeah, so vorinostat is an HDAC inhibitor, but really it's, the experiments showed its upregulation of GD2 and, in the studies. It was actually HDAC inhibition as a field. There's quite a lot of other agents that do the same similar thing, but vorinostat specifically had more upregulation of GD2, which is why it was chosen.
Okay, so y'all already knew that it wasn't an incidental finding, that
we knew some parts of it, but not really the GD2 upregulation. In fact, right now there's a whole project in the laboratory that is looking at other agents that could do the same thing, because ultimately, if we know that chemoimmunotherapy really works, we need to figure out ways to really upregulate that. So vorinostat is one compound. There are likely others, and there are others. It's just they're much farther in the laboratory aspect and not yet to the patients.
Okay, that's exciting. Thank you.
Okay, a question here.
Hi.
Oh, yeah.
This one's still not working. Thank you. I have two questions. Both are in regards to the lorlatinib and ALK portion of your presentation. My first question is, in regards to the patients that have a mutation versus an amplification, the response rate that you saw these patients responding to lorlatinib, is there a difference, or is it just ALK across the board?
So it's ALK across the board. We had very few ALK amplification patients, so I can't really comment too much about it. Some of our ALK amplified patients also have our NMIC amplification concurrently, and they are in very young patients, and those patients tend to have sometimes very aggressive disease. In this situation, we have too few in this specific study to comment.
And then in regards to the patients that are on those trials, whether it's through NANT and COG both, depending on what type of hospital they're treated at, when a patient is on a study taking lorlatinib and they're unable to follow protocol and they fall off of that study, is that positive data of a patient that's having a great response to lorlatinib still reported to you or COG in regards to helping patients in the future who are newly diagnosed and may be needing lorlatinib? Is that data still going somewhere to have a positive impact on these newly diagnosed children?
Yeah, very good question. Every time a patient enrolls on the study, that data goes forward and is included in the overall study. Now, there are some points when the patient comes off protocol therapy that we are restricted about how much more data we can then submit, but I think survival data and first relapse data is always included, no matter if they took the lorlatinib or not, or they took it, for example, on a commercial basis. And so we do try to gather that data, and there is no doubt that any patient enrolled is contributing quite significantly to the study. In terms of who gets the data, it's really the organization that's running the study initially until the study is a little more mature, and then it's then disseminated among the research groups. In our group, whether it's NANT or COG, there is a lot of crosstalk, obviously, but the data has to be mature enough before we can really disseminate it even wider.
Okay, thank you.
Hi, quick question. These NK cells that you talked about, do they require GD2 expression, or do they work on a completely different mechanism? I'm sorry if you said this during your talk.
No, they don't require a GD2 expression. However, NK cells are just there to, for example, we have NK cells even when we're fighting infections, right? They don't really care normally at what it is that they're going to try and kill. However, in this situation, they're only going to kill things that are tagged. In an infection situation, it's tagged by one or two other things. In this case, it's tagged by the fact that GD2 is on the tumor cells. But in order to see the tag, that's where the TGF-beta comes in. Learning, it's sort of growing up with TGF-beta around it, makes them better at seeing things, because one of our problems in neuroblastoma is that the neuroblastoma tumor cells then releases compounds that make the NK cells not as active. But if they're grown with TGF-beta together, then they're able to overcome that immune suppression.
But to see the tag, you still need GD2 expression.
Yes. They don't need to see, they need to see a cell that's abnormal. It's not GD2 expression, but it needs to have something on it that makes it abnormal. It's able to see it better when GD2 is tagged to it, but it doesn't have to.
Okay, so it helps. Got it.
One more question here.
My question is regarding the MFBG scan. We had we're actually part of the trial, and we're amazed at how detailed and how clear and how toxic it was. And in fact, we were able to get it a second time through compassionate use. My question is, how rapidly is that trial progressing? Do you see that as becoming a standard of care in the future? For us, it just seemed like a wonderful option. So
it's absolutely something that we're all asking about, how quickly this can become a standard of care. And I think the things that are the steps that we need to go forward with is, A, the FDA has to be convinced that it is something that you could use, meaning it is not giving false positive results, in addition to not having false negative results. So those are the two things that are going to be very, very important. That data is being worked on by the company currently, with the FDA. The other aspect of using it will be, what does it mean also? And that, what does it mean, is going to take a little bit of time, because yes, it's clear, it can be clear, but the question is, if you act on that, was that really something that you should have acted on? And that will only be answered by some of the studies that I think Dr. Pinto will probably talk about as well, that are going to go on, so that we can look at it and try and understand, was this a lesion that was actually going to progress or not? And maybe it's just something you see, and I think we all share the thought that it's probably going to be something that will get incorporated in the future. We just need to understand how to use it the right way, without, again, scaring people or making false decisions based on the scan.
Thank you. One last question.
If a patient has had a pharmacogenomics testing at the beginning of their treatment, does that identify what would work best for their specific genetic makeup?
Unfortunately, just like you saw in some of our earlier slides, neuroblastoma, or all actual cancers, evolve constantly. And so the data that you get at the beginning, while helpful, is not really the only data that one can rely on. When you're trying to make a decision on a relapsed patient, the biopsy at that time point, or in this case, the liquid biopsy at that time point, is really probably going to be even more informative than what it was at diagnosis, because this is an evolving process. How we make these decisions about treatment, right now, we obviously use some of our basic, the data of the historical data of how we have treated our patients, in addition to some of the evolving data that we are getting, and try to add our evidence-based scientific discoveries to the treatment. But I think at diagnosis, that is more limited than in relapse, where there's a lot more new data coming, and tumors evolving, and us being able to incorporate what we're seeing in our relapse to add to their therapy.
Do you think it would be beneficial to do a repeat pharmacogenomics testing after they've been through treatment and their DNA has changed?
I think depending on the goals, meaning where the disease is in the patient, especially in patients who are not responding as well, then repeat testing biological testing overall is very helpful.
Okay. And one last question. For patients that are not responsive to MIBG, would the MFBG be something that would be more promising?
So MIBG therapy is essentially radiation, right, linked to the MIBG, but the MFBG is really a PET imaging. So this is not a therapeutic.
That's what I meant. Like if a patient if they don't show anything on the MIBG, would the MFBG be more just better in general?
So think about an MFBG like a PET. Yeah. So yes, if your MIBG not added, that may be a scan, an MFBG scan may be helpful.
Okay.
I think therapeutically, how we treat the patients with radioactive compounds like MIBG, I do think in the next couple of years we'll be seeing different kinds of radioactive agents coming down the line that may also be able to access neuroblastoma in different ways.
Okay. Thank you.
All right. Thank you, Dr. Marachelian.